Hi, everyone. Welcome to the second webinar of this year's AGM Preclinical Translational Science webinar series. I'm Serene Markosian. I lead the Assay Guidance Manual program here at the National Center for Advancing Translational Sciences, part of the National Institutes of Health. We are super thrilled today to introduce our speaker, Dr. Lotte Bjerre Knudsen. Dr. Knudsen is an honorary professor in translational medicine at Aarhus University in Denmark. She is a former employee of Novo Nordisk, where she was last the chief scientific advisor for research and development. Lotte has deep experience in drug discovery and development. Her work has evolved much around GLP-1 based medicines, and she has been involved from early ideation back in the 90s, where she led the first program on a long-acting GLP-1 medicine, and all the way to recent work detailing mechanisms for this medicine in rodents and people with disease. Lotte has received numerous awards for the work on GLP-1 medicines. I'm just going to mention a few. She's the recipient of the 2023 Paul Langerhans Award from the German Diabetes Association, the 2024 American Association for Advancement in Science Bhaumik Breakthrough of the Year Award, the 2024 Lasker-DeBakey Clinical Medical Research Award, and she recently won the 2025 Breakthrough Prize in Life Sciences. Her contributions exemplify successful translational research, bridging basic discovery to transformative therapies for metabolic and related diseases. Her title today is GLP-1 Receptor Agonists, Mechanisms, Translation, and Therapeutic Impact. If you have any questions for Lotte, please drop them in the Q&A box, and we'll definitely get to them at the end of this webinar. And Lotte, on behalf of my colleagues here at NCATS, all of our attendees here today, we're super thrilled to have you, and the floor is yours.
Well, thank you so much for the very kind introduction, and I'm just so pleased to be here from my home office in Copenhagen. So I will add a few extra details to what you said, right? So these are my disclosures, and I am a named inventor of the first generation long-acting GLP-1, but I don't have any ownership. It's all fully owned by Novo Nordisk, and I have no other financial or any disclosures. I do want to say that, of course, the work that I'm presenting, I've been almost 40 years with the company. So the work that I'm presenting is obviously all done either within the gates of Novo Nordisk or in collaboration or funded by Novo Nordisk. So full credit to the company and the many collaborators for that. And I also want to say, it's my first time speaking as no longer being in the pharma industry. So I still have a little bit of a practice to do on when I say we or I or something like that. So forgive me for that if it sounds weird, right? And I want to say a little bit more. You mentioned these awards, and thank you for that. But I also just want to call out how committed Novo Nordisk has actually been to GLP-1 based research, and in particular for obesity. And there's a little bit of an extra funny story around the Bhaumik Award from the AAAS Science Award. This one I shared with Richard DiMarchi, and many times you don't really know what the methodology is for these awards, but actually science, and makes sense that science has been very transparent about what the methodology was. So they actually went and looked at the early patent application, and that was where it actually is evidenced that both I and the team I led at Novo Nordisk, we were focusing on obesity, as well as Richard DiMarchi, who at the time was head of research at Lilly. But still, even though Richard was head of research, and he has said this himself to me, he couldn't get Eli Lilly to prioritize GLP-1 for obesity. And, it was just, there's some good quotes from around that time, when people were saying, there's never going to be another medicine for the treatment of diabetes that's an injectable and is not an insulin. Well, I think we've proven them wrong. And even more conservative was the thinking around peptides in obesity, because people just didn't think that peptides could impact on the brain. And Svetlana Mojsov and Richard DiMarchi have both been in the lab of Bruce Merrifield, who got the Nobel Prize in 1984 for the peptide synthesis. So they are both telling me how many times they have heard this, peptides, they're not real medicines. I think we've proven them wrong. And I also wanted to say a little bit more about the Lasker Award. And this figure is from my JAMA viewpoint paper that goes along with the Lasker Award. And there's a couple of things I want to mention. I just want to mention, it's been so many years and so many people has been along on this journey. And that's really, successfully applied science is always the work of the many. And it's very complementary what people do in the industry as well as in academia. And sometimes, the science in the industry gets a little bit villainized in some way. And I think that's a little bit unfair, right? I think both fields are very productive. And sometimes I would argue, the work from the pharmaceutical industry is more reproductive, is one thing you could highlight, but they really work well together. And also it was very important for me for the Lasker Award to actually point out how is work in the industry, because when you look at some of my papers and you see me being the last author, for example, that doesn't mean that the first author works in my group. It might be an independent group at Novo Nordisk where I just, as a project leader, I make sure that everything is coordinated across. But it's different from many academic groups where you would typically say that the first and the last author comes from the same group. So I made sure actually, and there are 3 different inventors on the first generation GLP-1. I'm one of them. But there are 3 other inventors for the second generation GLP-1 semaglutide, and all of the names are written in. Right? And if you want to understand the story, so then read my viewpoint. Don't believe anything you read. Everything you read in the media, because very few people were around. The number of people interested and working with GLP-1 back in the 90s were tiny, tiny few. We were always at the back of the conference center in the smallest room. So we all knew each other back then. Now, of course, there's many more, but back then it was a very small field. And the viewpoint also goes a little bit into what is it that has actually happened. I think it's fair to call, especially GLP-1 for obesity, for an actual revolution. And when you look back in medicinal history, there are other examples where, for example, blood pressure lowering medications, cholesterol lowering medications, when they first were developed, invented and developed, they were first prescribed for the ones most in need, and then with time, when the patents ran out, then more and more evidence was apparent, then they started to get prescribed for more and more people. And that's also going to happen for GLP-1, even though we see now that not as many people can get access as wanted. So that's one thing. Another thing is that the dialogue around obesity has absolutely changed. If you haven't seen Oprah's program on obesity, that was about 2 years ago, I think it's worth seeing, because, she speaks to 3 different groups of people, those who really like the medicines, those who don't like the medicines, who don't want to talk about body weight at all. And she says, keep talking, but actually respect each other and listen to each other. And I think it's a good point, because it's like when it comes so close to the individual person as weight is, that it's like that some people get a little bit offended just talking about it. But obesity is a disease, and there's much more we need to talk about. And now that we have effective medicines, then often you can also talk more about the disease, and we also need to talk about the food industry. But let me get into the actual science, and I also just want to show this very nice picture from the Lasker Award, because Joel Habener recently passed. And Joel was he was at the Massachusetts General Hospital in Boston, right? And I know he was a mentor for many people that has come out of lab, an absolute giant at the time who did some of the early work on identifying GLP-1, just like Juul Holst here in Copenhagen, where I am. So I have some take-home messages that I'm going to mention up front. So I think what has happened here, and I got a little bit to that already with the comments from Richard and Svetlana. That these peptide-based medicines have absolutely been unlocked as a modality, and now everyone is looking at peptides, and in particular this fatty acid acylation technology. And I think also the work that I will show today also has changed how peptide-based medicine can impact the brain. Second point, the GLP-1 biology is broader than any other medicine. And GLP-1 based medicines, they often have effect on cardiometabolic diseases via separate mechanisms, and many mechanisms are often involved in in individual endpoints, and I'll talk a lot about that. And in some ways, you could say that the guidelines for multi-biology drugs should change, because the system doesn't really fit with something as GLP-1, and there are no other biologies that are that broad. So you're actually forced to say, okay, this is the label for diabetes, and then you're not allowed to mention that weight loss is a desired pharmacological effect, right? And that doesn't make any sense, right? And then towards the end of the talk, I'll get a little bit into what are my collective learnings around drug discovery and translation and development. Right, so how did we actually get to where we are today? Well, it's a long and winding road, and there was a lot of things that needed to be done. But let me start with a little bit of chemistry. Right? I started working on GLP-1 in the early 90s. In the beginning, I was actually doing a little bit of chemistry, but then soon I started focusing on the receptor pharmacology. I was in a group that was led by Ole Kirk at the time, and we were a lot of people from the technical university. So we were just starting to think about, if you were to make a different drug molecule based on GLP-1, what could you do? Where in the molecule could you actually make changes, and where should you stay away from, which is the red positions noted up in the upper left corner. We also worked closely with Juul Holst at the University of Copenhagen, so we learned that this is a very unstable peptide. It's degraded by DPP-4, and it has a half-life of less than 2 minutes. So then we started thinking, what about just stabilizing against DPP-4? And you could do it, and you could make potent molecules. That's what's shown on the left, but you only get a half-life of 4 minutes. So that was 2 years of work went down there, and then not very useful. And also in the late 90s, I was involved in a program led by Jesper Lau, and where we were working together with a company in La Jolla that doesn't exist anymore, called Allenex at the time, where we found small molecule agonists. And this is a nice paper of mine from PNAS in 2007, which I submitted without having a sponsor. And it's just these molecules were super interesting mechanistically, agoallosteric modulators, but they were not sufficiently drug-like. And actually later studies have shown that they covalently bound to the receptor. So that was at this point in time. Now we're in the mid-90s. There were already 3 failed projects at Novo Nordisk. So then in 1994, I became the project lead for the research activities. I was still kind of like fairly young, and I was also given an extra challenge that I only got 1 year to solve it, because now we already had 3 projects that failed. So I was sitting around thinking, what could I do? And then I decided to focus on this fatty acid acylation technology, which was around in the company at that time. There were some early studies where it was tried on insulin. So I said, okay, I think I could actually lead that. So let's try that. So the principle is simple, that you attach a fatty acid to the peptide, and then that binds to albumin, and albumin has a huge reservoir for binding of fatty acids, because lipids are, of course, insoluble in blood. So that's a physiological mechanism. And so when we had a very strict process for designing a drug, high potency, long half-life, high homology to GLP-1, high stability, we didn't actually need DPP-4 stability to make a once daily, which was the goal at the time. In the little figure, you see that's the GLP-1 receptor. This is a G protein coupled receptor of the B family, a fairly large extracellular portion. And then in red, you can see the little fatty acid. So it's in some ways, a very simple molecule. Sometimes I make fun and call it a steak, because it's a lipid and a peptide, which I guess is a protein. But of course, it was really complicated to find it, even though it looks simple. And also, this was the first time that the fatty acids was actually moved inside in the middle of the peptide chain. Before that, they had only been on the C terminus or the N terminus. So this was the first breakthrough. This became the first long-acting GLP-1, the first GLP-1 to be approved for obesity, and the first GLP-1 to show cardiovascular benefits, and actually also to formulate many of the hypotheses that were then later confirmed with the much more efficacious second-generation GLP-1. But what's really interesting about the chemistry of this class of molecules is how different they are. So in the lower part of the slide, you see the size of the molecules, going from now small molecules to really large towards Fc fragment molecules. But what really stands out on the efficacy side is how different they are when it comes to weight loss. So exendin 4, which was isolated from the saliva of the Gila monster, was the first approved GLP-1 for diabetes in 2005, but it didn't have much weight loss. Neither did the first once-weekly molecule, dulaglutide, the Fc fragment, also doesn't have much weight loss, never tried in obesity. And it was then semaglutide that was the second generation GLP-1 from Novo Nordisk, which has a much different fatty acid and spacer, a much more sophisticated molecule in a way that really has set the efficacy apart in the class. And with the new approved dose of 7.2 milligram, it passes 20 kilogram weight loss in people with obesity. Now we also have orforglipron, so a small molecule approved. This molecule was originally identified by Chugai in Japan, and then Eli Lilly picked it up. But it doesn't have the same weight loss as semaglutide. So it's really important to look at the doses in order to, and the different molecules in order to understand the pharmacology. And then with semaglutide, there's another unique thing, and this is from a paper where I'm the last author in Science Translational Medicine 2018. There are 3 shared first authors, all from Novo Nordisk. It's years and years of work of actually figuring out how did we actually make an orally available formulation. So with semaglutide, you have a once weekly injectable, but you also have a once daily oral. So here's a graphic of the tablet, where inside the little tablet, you can see the real size relationship between the enhancer snack, originally identified by the company Hemisphere, and then you can see the structures outside the tablet. It's a lipid, it's a small lipid-derived molecule. And the ratio between the molecules is approximately 500 snack to 1 semaglutide. And then the reason why we can get it to work and semaglutide isn't degraded is because the majority of the absorption happens in the stomach. So when the tablet lands there, snack helps to form a small microenvironment with increased pH so that semaglutide doesn't get degraded. And then it gets closer to the cells, and then the absorption happens with a transcellular process where locally the mucus layer is detached, probably the same mechanism that happens with ethanol, just as a common reference, right? And then you have the absorption taking place. Of course, this is much more inefficient than the subcutaneous injection, but it does work, right? And you can get the same kind of exposure. So how about the biology? This is where it gets really, really interesting. So the GLP-1 physiology throughout the body is, of course, governed by where the GLP-1 receptor is expressed. It's a G protein coupled receptor, B family. There are no subtypes of this receptor, but it's distributed on very specific cell types, but many organs, but very specific, coupled to this physiology. You know, it all has to do with what needs to happen in the postprandial situation, everything that needs to happen after we have eaten a meal. And in physiology, it's secreted from the L cells as well as the hindbrain and the NTS. But that doesn't have much to do with the pharmacology, because the amount of GLP-1 that's secreted in physiology is very small compared to the pharmacological levels that we have 24/7 every day. And so this is the biology, or just some of it, the ones that are most well described. So I like to say that GLP-1 has multifaceted system-wide effects. And I'm not going to talk much about diabetes today. That's the most well-described area. But importantly, sometimes I hear people think it's just about insulin secretion. No, it's not. It's also just as important that GLP-1 lowers glucagon secretion, and it has a minor effect on gastric emptying to reduce postprandial glucose. When we're talking about cardiovascular benefits, then it's both inflammation, direct effects on the heart, triglyceride lowering, systolic blood pressure lowering, and so there are always many mechanisms involved. And let's dive into some of them. So here's something around the GLP-1 receptor, working towards making medicines and describing how they work, and also sometimes how they don't work when you have to figure out if something is a side effect or not. It's so very important to know where your target is expressed. G protein coupled receptors can be really tricky. And it's very easy to buy antibodies that are not specific at all, and then you're just not looking at the GLP-1 receptor. You can always look up Dan Drucker on Twitter. Sometimes him and myself, we call ourselves the receptor police for pointing out where people are using antibodies that don't really recognize the receptor. This is from a paper of mine with my colleague Charles Pike, who is the expert at Novo Nordisk that's doing all the immunohistochemistry, either himself or in his team, right? And I'm the last author on this paper. So interesting to the right, you see renin-positive smooth muscle cells in the juxtaglomerular artery in the kidney. There's actually quite a few places where smooth muscle cells, kind of like around the entry to organ, has the GLP-1 receptor. So I think it's maybe something with actually a local organ protection and maybe, stimulating blood flow and oxygenation of the tissue. And another one I find really interesting is up in the upper left corner, the Brunner's gland, which are mostly known from cystic fibrosis, or maybe you don't know them, but I can tell you that people with cystic fibrosis, they also have really bad mucus in their intestines, and that's because they have the chloride channel sits in the Brunner's gland, and it means that the mucus secretion, which comes from the Brunner's gland, is too sticky, right? But these glands are filled with GLP-1 receptor, and GLP-1 stimulates mucus secretion. It makes sense. You think you've eaten, the food will come down into the intestine and the stomach. It makes sense to stimulate mucus to protect the intestine. It's unclear how it relates to different outcomes, but it could have something to do with actually making got the barrier function better, and that's a way to lower inflammation. And also in the brain, right? Of course, there are lots of neurons that express the receptor. And this is from a Shiny app from Stanford. I don't think it's published in a peer-reviewed journal, but it's a very useful data set. Again, showing that. So these are non-neurons, right? Again, it's the vascular smooth muscle cells and his astrocytes. So maybe again, something around oxygenation of brain tissues. But so now I'm going to skip into like a section around sort of like a many high impact clinical data in people with diabetes and obesity, and I'll focus on some of the mechanistic learning. So I'll flip a little bit through the actual outcome data and then say a little bit more about the mechanistic learnings. So I mentioned liraglutide became the first GLP-1 to show cardiovascular benefit. If you were around at the time, you know how controversial it was. People were starting to think that it was actually dangerous to lower blood glucose. So it was a surprise. And the year before, in 2015, we had the first cardiovascular protection data with an SGLT2 inhibitor, and then with GLP-1 in 2016. I wasn't surprised because, we had lots of biomarker data. So on inflammation listed here on the lower part, but also triglyceride lowering, systolic blood pressure lowering. It was natural to expect that it would actually work. And there's even a 15% reduced risk of death from any cause. So these results, because we were starting to think more and more at Novo Nordisk that it wasn't glucose lowering or it wasn't weight loss. It was something else, and maybe it was inflammation that was really important. So this actually went into the thoughts to design SELECT, and SELECT is a landmark study because it's the first time ever that anybody has shown that you can lower cardiovascular risk in people with obesity with a drug, and specifically semaglutide. And again, death from any cause is significant. I spent many years trying to convince cardiologists that GLP-1s would be doing this to an effect on atherosclerosis. This paper in JACS from 2018, I'm the last author, but again, the Gunnar Rakopolsky, who is the first author, this is really his work, right? So what we did here, and I should also want to give credit to Anthony Deere in Australia, who was the first one to suggest in animals that GLP-1 stabilizes atherosclerotic plaque. So what you see here is some semaglutide data in LDL receptor knockout mice with 3 different doses, where the highest dose with the purple color has a lot of weight loss. The green dose has almost no weight loss. And it's very consistent with the literature that you can see effects to dampen inflammation at quite low doses of GLP-1. It's not an anti-inflammatory medicine because that's a mechanism that blocks IL-1 or IL-6 or a particular molecule. I think what GLP-1 does involves many mechanisms, and so I like to say dampened inflammation, or maybe induces a new level of homeostasis in the system. But in people, I think that means increased stability. And out to the right, there's a clinical study. It's not a randomized controlled trial, but it is 200 people showing a large increase in the fibrous cap. with liraglutide. And then let's stay in the cardiovascular system and talk about HFpEF, as it's called among friends, heart failure with preserved ejection fraction. And really strong outcome data. One of the key endpoints in heart failure is 6-minute walking test, and it's increased with 20 meters. And it might not sound like a lot to you, but if you have heart failure, this is really significant, and a 40% reduction in inflammation measured by CRP. But what's really interesting is that people thought GLP-1 would never be an option for heart failure. And because we were the first ones to ask, now I said we, right? At Novo Nordisk, we're the first one to ask for a cardiovascular indication. And because of this concern, right, and then the concern was that there was an increase in heart rate, about 2 to 4 beats per minute. And then people were saying, based on historic data, that's linked to an increase in CV mortality. So we thought, why don't we should look at the mechanism. So again, back to that histology and actually understanding where your GLP-1 receptor is expressed. And my colleague at the time class, Charles Pike, him and his group, they sliced up an entire non-human primate heart and found the GLP-1 receptor only expressed in the sinus node. There's still a little bit of unclarity, I think, in the literature based on human data whether other cells express the receptor, and it's a GPCR. They can be lowly expressed, but this is a very good explanation for the increase in heart rate. We also started to do genetic studies. Novo Nordisk has a team in Oxford. The people who did this study up in the right, Dipender Gill and Joanna Hawson, they're not there anymore. But if you want to learn something about Mendelian randomization, look up Dipender Gill. He has hundreds and hundreds of studies. And the strongest trait that GLP-1 is associated with is actually prevention of heart failure. So it's just a nice reminder to think, go out, understand the mechanisms before you start saying whether something might work or not. And here's a paper from 2030 in a mouse model from a Dutch group that's in Groningen or Erasmus Rotterdam. I am a co-author, but again, it really is their work. So they have an angiotensin II induced model of HFpEF, heart failure with preserved ejection fraction, kind of mimics what you see in people. So with semaglutide, you see an increase in exercise capacity. You don't see that with pair feeding. And also they see a significantly less fibrosis, with no difference in cardiomyocyte stiffness. And I like to point that out because, again, people have been saying, GLP-1 doesn't have an effect on fibrosis, but a pattern is starting to emerge that it actually has. Again, that doesn't mean it's an anti-fibrotic, but amongst the many mechanisms, and maybe there's something with endothelial cells, it looks like it also can have an effect on fibrosis in some tissues. And here's the kidney outcome data, and again, benefits, including cardiovascular death. And there's, and this is even with a fairly low dose of semaglutide. And there's some very interesting work ongoing right now with mechanistic studies. There's a small study that has been shown at conferences, but is not published in full yet, where biopsies were taken and single-cell transcriptomics and spatial transcriptomics have been analyzed from patients in the control and the semaglutide group. So I think that's probably going to come out this year or next, right? And that's going to be very interesting to inform about the mechanism. And let's skip to the liver, right? And I apologize for the text in the bottom of this slide, but these endpoints in MASH are so complicated, and there are so many abbreviations. So I think it makes sense to put them in on the slide. But also here, we see an improvement in liver fibrosis. And then to another paper of mine where I'm the last author, but again, I think we have 25 authors on this, and this is really a collaboration between some people from academia, some people from Novo Nordisk, focusing on various different data sets. Here we really try to understand how semaglutide impacts MAC, and this is a classical mediation analysis shown here. So if you look in the top pink frame, you see the mass resolution. So this is the steatotic effect, driving fat out of the liver. This is clearly weight loss mediated. Look at the bottom pink. This is the improvement in liver fibrosis. This is not weight loss mediating. always multiple mechanisms via different mechanisms. There's a lot of data in this paper, so if you're interested in mass, it's a treasure trove of things you can look at, which protein are involved with the different outcomes. We also look at some specific proteins of interest in MASH or MASH, right? And we see a pattern where the ones that go up with semaglutide go towards a healthy level in an independent cohort, and the opposite with those that go down with semaglutide, they also go towards the healthier level. Right, so now I'm getting to the brain, and I think it's fair to call maybe what has happened with obesity and GLP-1 for the most impactful area of everything that GLP-1 has shown up. Obviously, these medicines are super important for people with diabetes, with effects on so many different comorbidities, but there were no effective medicines for obesity before, so it's really there that you have the biggest impact. And sometimes I hear people saying, it was all a chance discovery. Nope, it was not a chance discovery to take GLP-1s into obesity. That was a strategic decision based on the science at the time. This is a paper from Nature in January 1996 by Stephen Bloom. Animals dosing GLP-1 directly into the brain can make even starving rats completely stop eating. Binding sites in the brain, C-FOS activation in the brain. At the time, I was leading the program that led to the invention of liraglutide. The world was a little bit smaller back then, so I was actually inspired by another group that was working locally in Copenhagen, led by Ole Madsen. He had a different animal models where there were high levels of GLP-1 and glucagon and PYY, and those animals starved themselves to death. So, I was just thinking, here I am leading a program in GLP-1 for diabetes. Why should I not propose, because many people were looking at obesity, that we could take it into obesity as well. And I didn't realize at the time, no one told me how provocative it was to take a peptide into obesity. And we just followed the data. And a few years later, there were also data in humans showing that GLP-1 could reduce energy intake. So over the years, we worked a lot on the mechanism because, when we had to get liraglutide approved for obesity in 2014, it was so controversial whether we should even have more medicines for obesity. And FDA gave us an advisory committee. I've done 5 FDA advisory committees, and we're the only company who's gotten them because we were again, I'm saying we, Novo Nordisk were the first to do everything, right? The first with a long-acting GLP-1, several safety issues that had to be settled. The first in obesity, the first with a cardiovascular indication, the first with the second generation, much more efficacious GLP-1s. So understanding the mechanisms, particularly in obesity, because people were saying obesity medicines, either they have cardiovascular side effects, or they don't work, or they have suicidal ideation. And worth noting that FDA has actually just removed the suicidal ideation labeling from the GLP-1s, right? That's really rare that happens. But at the time, it was controversial. So this is a summary of these particular data are from the JCI 214 paper. So there's a lot of GLP-1 receptors in the brain. And if you look at that to the right, and this is a technology developed by the histology team at Novo Nordisk with whole brain staining and then single plane illumination microscopy. And if you look at the liraglutide pattern on the right, you see, then the conclusion is that GLP-1 does not cross the blood-brain barrier because there are so many receptors where we don't see any binding. But there are many receptors in the circumventricular areas, and those express GLP-1 receptors. And if you look in the middle picture, you see in blue, you see C-FOS staining, and the yellow staining is a downloaded experiment from Allen's Brain Atlas that tracks the lateral parabranchial neurons that are sometimes called, the highway of meal termination. So it's likely, or it seems to strongly indicate that the signal to the amygdala, for example, goes through there. So GLP-1 acts on energy homeostasis by orchestrating numerous directly and indirectly targeted neurons, and the effects include both homeostatic and hedonic mechanisms. And sometimes I see in the mouse literature, that people are saying, oh, this particular neuron is necessary. And you can say that with genetic ablation, but in pharmacology, I think they're all involved, right? So I think it's a little bit of a conflict between the pharmacological literature and the genetic model literature around the vocabulary used. A quick shout out to, and this is from the DCI Insight paper in 20 that focuses on semaglutide. Again, there are 3 shared first authors, and I'm the last author. And just the septal nucleus is an area that really discriminates where semaglutide has much better access. There's a lot of GLP-1 receptor expressed on terminals in that area, and it's not really well described exactly how, there are well described, of course, connections and projections to the rest of the brain, but again, how it all plays together and not really known. But it's a good hypothesis, at least one of them, for why semaglutide gives more weight loss than liraglutide. So a quick overview on clinical and scientific combined mechanism understanding. Sometimes, again, I hear people say you don't know how these medicines work in people. That's not true. We know they help people lower intake, and they make people feel less hungry, more full, more satiated. And we have a good understanding of some very classical neurons in the arcuate nucleus in the hypothalamus. So POMC card neurons have GLP-1 receptors, they're activated. NPY GLP neurons do not have GLP-1 receptors, but they're still targeted, and we have a lot of EFIS data in the 214 paper that goes towards this. And then also when you ask people, and you get all these effects on the hedonic system. I do have a fun animal study that I didn't include here, where we gave rats access to different kinds of candy, and that was published already in 2007, where we saw that they actually changed preference, so they eat more chow, less candy. But much better than that, we now know also in people that people feel that they are less likely to respond to cravings, and especially if it's savory food. And because it's not significant any longer at 2 years, doesn't mean that it's not still numerically working. It's just not statistically significant anymore. And again, this thing about suicidal ideation, people actually report a positive mood when they're on these medicines. So my last section is around newer, before the drug discovery piece, just a few slides on newer methodology for further understanding brain mechanisms. There's so much going on in the brain with GLP-1. I think we have barely scraped the surface on what opportunities there are. So this is a Nature Medicine paper from 2025, also where, but a different one than the other one, where we focused on proteomics from the large phase 3 clinical trials with semaglutide and obesity. And then we compared proteomic signatures to the DCODE data set with disease signatures. And I'll show the full one on the next slide. But it's just very interesting how much real-world data there now are on these medicines to work against alcohol use disorder, opioid overdose, hospitalization for substance use disorder. And of course, there are no large, nothing is approved yet. But the FDA changed last year that now they're only asking for 1 trial for additional indication. That's really important because sometimes it can be difficult to make. Before that, they said you had to have 2 separate trials in order to get an approval. But let's just look at the blown up version here, right? It's just really interesting how the plasma proteomic signature points toward brain diseases. Obviously with BMI, sleep apnea, sleep insomnia, nicotine dependence, neuropathic pain, alcohol dependence, rehab admission, even depression and bipolar disorder. So I think there's a lot of things going on here. Obviously, this is not causality, but it's a really good hypothesis for looking further at whether these medicines could help more people with more endpoints. We don't have a lot of data from the human brain with these medicines, in obesity, or diabetes, right? There's a lot of human brain proteomic transcriptomics data in the Alzheimer's field, but there's not a lot in obesity. And this paper from Nature 225, which is from the Yo group in Cambridge, UK, Bruning group in Cologne, Germany. I am a co-author also, along with a few other people from Novo Nordisk, because we were much involved in discussing this study. So this is the first hypothalamic single cell and spatial transcriptomic study. Then there's also data sets coming from the hindbrain. I think this is super important because much of the data that we have been based on mice, and we need to understand much better what exactly it's like in the humans. In the middle lower part of the picture, you see the GLP-1 receptor and the 5 different neuron subtypes that have the GLP-1 receptor. There's also 5 that have the calcitonin receptor, which is a target for the amylin calcitonin ligands, which are not approved yet, but have been submitted for regulatory approval, the first one. And there's very little overlap, speaking to the combinability of these 2 kinds of medicines or drug candidates for the treatment of obesity. Another way to get human relevance into your system. This is from a Nature Metabolism paper from 2021 from the Tune Pears group in Copenhagen, Myers, Michigan. Again, I'm a co-author. This is focusing on the hindbrain and looking at, if you look in the hindbrain, you see which neurons are enriched for human BMI-associated genetic variants. Then you find the GLP-1 receptor and the calcitonin receptor and one other subtype. So that leads me to my last section, and it will just be a minute I'm going to spend on this. So we should have plenty of time for questions, which is always so very interesting to get questions. So what has all of this, these 35 years in the industry, taught me about drug discovery? With GLP-1, we had human data early on, but we also went into other indications where we didn't have human data. And there's also, I could have also spoken about how there were lots of things even around diabetes where we thought that GLP-1 would have really strong effects to increase beta cell mass that didn't turn out to be true. So I also have some learnings that it's really important if you use animal models that you should know how exactly they reflect the disease you're looking at. But if you want to do drug discovery and if you're looking to do something that hasn't been done yet, focusing on a biology where you don't know the translation, you really need to understand both the molecular level, the cellular level, and the physiological level. Because if you don't, you won't be able to make sure that you have designed your clinical studies in the right way. And you might also not be able to explain whether something is a side effect or not. And that's just very important, right? And, looking in the middle panel, you should also not fall in love with your target too early, right? Because it might not be the right one in the pathway. It might just because you have the biggest genetic size or the biggest effect in an animal model doesn't mean that it's the best target to drug. And then, of course, there's the actual druggability. Where's the target expressed? Are you actually able to pair it off with a good drug modality? And unless you have endless funding, it's a good idea and endless time, and nobody has that, to look at human relevant data. Genetics is really coming of age, so you can look, well, you should look both at common and rare variants, but we also need much better data on directionality and mapping them to an organ. It's really difficult, especially within the brain. Of course, you should never look at genetics just in isolation. It should also be always be part of the combined picture. And nowadays, there are many human cellular intervention data, right? And you can also work with real world data, which can be very informative sometimes because there are drugs out there that have effects on different pathways, just like GLP-1. But then here's the thing, right? AI is coming in all over. And of course, AI is no magic solution, but it is really speeding up some areas significantly. And it's something you need to be thinking about, right? Because, the next breakthrough innovation might come from the tech industry. And, there are really, really smart people out there like Patrick Su at the ARC Institute. In San Francisco that are creating many of these, some of these newer foundation models like the EVO1 and EVO2, and I think he's probably have an EVO3 coming as well. And he has some really interesting thoughts about cellular resistance and how you can put that into AI models. And then, of course, it's all about these agentic AI models that are now becoming much, much easier to make. And if you're not using Claude, you probably should get started on it. Right. So is again back to my take-home learnings. We've really unlocked peptide-based medicines as a modality with this fatty acid acylation, and the view on how peptide-based medicines can impact the brain has significantly changed. The GLP-1 biology is broader than any other medicines, always multiple mechanisms involved in the different outcomes. One should think about whether the guidelines should change because of that. And if you want to make breakthrough medicines, you need to understand your mechanism of action and your physiology. And then, of course, collaborate, because that's really how how you get things, how you learn best. And the more people you talk to and ask them to give you feedback, ask them to grill you and give you critical feedback, because that's just so important learning. And you should be really thankful for those people who actually does criticize you, because that's so valuable. And you can connect, if you're on LinkedIn, you can connect to hear my views. I tend to have a lot of them, and I will always respond to people, maybe not the same day, but I will respond. And thank you so much for your attention.
Thank you so much, Lotte, for this amazing presentation. And I would also like to thank you for all your efforts to actually bring this medicine to the people in need. And I'm sure everyone here shares this sentiment. All right, so let's start with the questions. We have multiple different questions. So I'll start with one here. So the question asked is, I understand that many immune cells express GLP-1 receptor. What are the prospects about the role of GLP-1 agonist in immune or infectious diseases?
Yeah, so thank you for that question. I like that really, because I'm not sure that many immune cells expresses the GLP-1 receptor. And I didn't have time to speak about that today, but I think, I mentioned the Brunner's gland. I think it's really important. It dampens inflammation coming in from the intestine, right? And then there are some neurons that have been shown within the last couple of years. There's been two big papers pointing to neurons in the hindbrain that have really important effects. Again, it's in mice, to control both central and peripheral inflammation. I think that's really important. Macrophages, I don't think they have the GLP-1 receptor, but they do get reprogrammed. So I think probably from a neuron, again, in the hindbrain, to then be have a less inflammatory state. And then there are those now famous T cells that got the Nobel Prize last year. Actually, sometimes when I present, I do have a slide with the T cells, because it looks like they have the GLP-1 receptor. So when I listen to the Nobel presentation, they said there was no therapeutic impact yet, but I do think GLP-1 has a little bit going on there. So I think that's the point about the GLP-1 receptor in immune cells. And then, but the result is that there's up to a 50% lowering in CRP, but it's not a mechanism that goes in on one pathway, but it's a lot of different pathways. And the data I showed you from the plaques in the animals, there's no GLP-1 receptor locally in the tissue on any of the immune cells. And that's also very interesting about, you meant the question, the person who asked the question also asked about infectious diseases, right? There's not good clinical data here, but it's worth pointing out, right, that the cardiovascular data shows no increase in any other death or risk from infectious diseases, just like was shown with IL-1, right? So GLP-1 has this powerful reduction in CRP without an increased risk for infection. And also remember also the COVID data, right? The people who were on GLP-1 based medicines were protected partially from death, right? Had a better outcome.
Wonderful. Thank you. So the next question here is, what long-term effects might we expect to see in healthy individuals without diabetes or obesity who take GLP-1 medication? Could they be at higher risk for malnutrition, organ failure?
Yeah, that's also a very common question, and of course, people shouldn't take medicines they're not indicated for, right? That would be my stand, We've worked so many years hard to prove how these medicines work and show the benefit-risk ratio. So I don't think people should be on them if they're not indicated, right? But that being said, I don't think there's a huge risk, right? I think the majority of people who take these medicines, they really need it because they have either a BMI over 27 or even much, much, much higher, right? And there's no really good data I can point to because, of course, there are no studies like that because people shouldn't be on them.
Understood. Okay, next question. What kinds of cell considerations for certain cell types come to mind or are somewhat obvious to you? I'm not well versed. And are there any virtual cell models that are particularly interesting to you?
Well, T cells would be interesting, right? uh, um, so what, but it's the brain ones. We don't really. I think there are cell models from the POMC card cells that's beginning to be available, but there are not a lot of models available for some of the other cell types in the brain. So it's really the brain that is the most unexplored organ for GLP-1. So also at this point in time, there are so many good hypotheses and based on real world data. So it's for GLP-1, I'm not really sure we need the AI models. This was more something I said for other drug discovery efforts with novel targets where you can learn how big is the effect size if you compare different targets.
And just to follow up on that conversation regarding the brain, so there's a question here. So the CNS proteomic slide would suggest that a small molecule GLP-1 agonist capable of crossing the blood-brain barrier might open up all kinds of neurological opportunities.
So the current small molecule orforglipron that was recently approved doesn't cross the blood-brain barrier. It has a molecular size of 840 something, I think, and it has a physical chemical properties that means it won't be able to travel in the brain parenchyma. So there are no small molecules for the GLP-1 receptor currently that can deep penetrate the brain. And those images I showed with where you see binding and activation, it looks exactly the same with orforglipron.
All right.
There are people, of course, you could think about, there are more GLP-1 receptors. Could you target that in some way? It couldn't necessarily have to be small molecules. It could also be with a brain shuttle, or you could get more GLP-1 into the brain. But then, of course, that's a completely new benefit-risk ratio, because, of course, you could get more efficacy, but you could also get other things you just don't know yet.
Makes sense. Thank you, Lotte. So also just a reminder, if you have any questions, just drop them in the Q&A box. That's so we can get to as many as possible. Lotte, there's plenty of questions, so I'm going to go through them and we'll try to get as many as possible. All right. So here's another one question from a small molecule chemist here. How do the highly plasma protein bound lipidated molecules with long half-life interact with the receptor? How is the steady supply of free fraction regulated, and what is the main mechanism of eventual clearance?
Yeah. So in general, right, and it's a little bit different for the once daily versus the once weekly. So they bind to albumin, right, and then that's just an equilibrium, right, and the majority of the molecules of the amount is bound to albumin. So for semaglutide, for example, it's probably around 95.5% is albumin bound, and then as it gets cleared, which it does by clearance partly in the liver, partly in the kidneys, then of course a little bit more will be released. But the vast majority is bound to albumin and is restricted to the plasma compartment via that binding to albumin. The small molecules are, of course, different, right? But again, as we just talked about, they don't deep penetrate the brain or other tissues. So they're also in the plasma compartment.
Understood. So the next question here, when GLP-1s were developed for obesity, there was a fear of being another rimonabant type story. Yes.
Rimonabant.
When rimonabant was pulled out from the market, this is a long question, so I'm just going to try to read it. From the market due to neuropsychiatric side effects, including risk of suicide. This was even more a concern given some reports from Iceland. We know now that is not the case, which is great. What is your take about how these drugs may reduce appetite and food seeking and possibly beyond food, example, alcohol? And yet these medications don't have such side effects, anhedonia, example. And if anything, there is a potential to help some people with depression. Do we know the mechanisms, how these medications have these meaningful effects without neuropsychiatric effects?
Yeah, so rimonabant, for if you don't know, right, was built on the cannabinoid biology. So it makes a lot of sense that there's a link to both mood as well as appetite, right? Maybe people dare admit that they even have personal experience with that. But this is where I mentioned, that we put a lot of effort into describing the mechanism. When we, like in the hypothalamus, it's very specific to the POMC card neurons and the NPY-AGRP. Then there's a whole bunch of other neurons in the hindbrain. There's a septal nucleus, but there's not this link to a specific biology that we know is involved with mood, right? But of course, I cannot stay here and explain, okay, so we get this neuron going to this neuron to this. I do have a slide on that, right? But it looks very complicated because there are several entry sites where you have the GLP-1 receptor being activated, and then you also have secondary activation site, and then it all kind of plays together, and probably executive function is probably also changed, right? And so I would say we know the mechanism, right? Because we know a lot of the receptors involved, and we know how this receptor type is activated. But can we explain in detail, so what exactly does the amygdala activation do compared to the septal nucleus, compared to the NPY-AGRP neurons? No, we can't, because they all kind of like end up being regulated by such a complex interplay of neurons and maybe even other cell types in the brain. But it's just a fact that we know, because there are millions and millions of people treating with these medicines, and we have close to 100,000 people, if not more, in total in large randomized controlled trials. So it's just a fact that we don't have any undesirable effects on mood, and actually even pointing towards the opposite, right? And how often does FDA remove a safety signal? That's really rare.
Thank you, Lotte. Next question here. Great work. What adverse events associated with semaglutide are most serious? Per Samantha's question, are there adverse effects that are implied but as yet undocumented that we should be looking for? So I think you sort of answered this, but
that's a very big question. Maybe I actually should have mentioned, there might be newcomers, right, that the most common side effect is gastrointestinal, nausea, vomiting. It's mostly mild and goes away for most, but there's some people who don't want to be on it because of that. So it would be great if you could find other medicine that don't have this effect of all. But there's very well documented that some of the neurons in the hindbrain also express pharmacological signals that are known to cause nausea. But those are the same neurons that actually are involved in lowering inflammation, right? So you might not be able to separate the cardiovascular benefits from the slight nauseating. So those are the most common, sort of like to go in and say whether there are any side effects that are undocumented. That's almost an impossible question, right? But I just want to say, that this is such a well-studied group of medicines, approved now to 20 years, and so many large cardiovascular and kidney outcome studies, and showing reduced risk of deaths from cardiovascular disease, deaths from any cause. And so it has been looked at multiple times, right? And the benefits far outweigh the risk for this class of medicine. But all medicines have side effects, right?
Thank you, Lotte. All right, next question. How does serotonin play a part in GLP-1 release? Increases in serotonin can increase natural GLP-1 and increase satiety. I believe that's what they mean here. Do the artificial GLP-1 also increase serotonin levels? And how do they increase with SSRIs, which decrease recycling of serotonin? I don't know if you got that question. But...
Yeah, so so here's so when it comes to in some ways I'll say like I'm not able to give a really good answer, but it's true that there's something with serotonin and receptors and GLP-1 release, but that doesn't have anything to do with GLP-1 receptor agonists, which are dosed pharmacological at 100 times higher levels. So there's no relevance there. And then specifically, and what was the last part of the question again? Can you just read that again? Was it something with serotonin signaling in the brain also? Or
no, I think the last part was how do they interact with SSRIs, which decrease recycling of serotonin?
I have to plead the fifth on that one. I'm not sure I'm able to give a good answer.
No worries, we'll move forward. All right, there's plenty of questions, Lotte. So
there's a couple of people also who have raised hands. I don't know if you want to give them a chance as well, or
we don't usually, but I don't mind. Let's go ahead.
All right.
So Sita, go ahead, please. Abigail, can we unmute Sita so he can ask a question?
Oh yeah, they have to be unmuted. Yeah, you've blocked them all.
Let's see. I think it might work. Lotte, you give us a second.
Can you hear me?
Yes, go ahead. Sita.
Oh, thank you so much. I didn't have any way of putting the question on the question and answer. I didn't have that option,
No
but
worries.
thank you, Lotte, for a fantastic presentation today. As a person who originally worked with Richard DiMarchi on some of these projects, it was really great to hear what you were talking about. It was wonderful. I have a very fundamental question. This medication that has such fantastic effects on multiple organs in the body, and it seems to be almost a miracle medication. My question, a fundamental question, is that why is that the native molecule, when it's secreted, the nature has evolved that into be such a short-lived molecule? And you guys had to figure out a way to extend the lifetime of the molecule to have all these fantastic effects. So I was kind of wondering why nature had this way of producing such a molecule in such a short half-life.
Yeah, great question, right? So in physiology, right? So GLP-1 has all of these effects. I mentioned I had that wheel of fortune almost with all the many effects, right? But of course, it's part of the postprandial bucket of things that need to happen, and GLP-1 is not essential in that, right? There are hundreds of peptide hormones and neuromodulators that are also involved in similar processes. And if you look at the GLP-1 receptor knockout mice papers, you might fall asleep reading them, right? Because there is a relevance when you knock out the receptor, but it's a tiny one. So sometimes when I speak, I also speak about saying if GLP-1 had come from the mouse side of things, it would have never been taken forward, because people would have thought, oh, this effect size here on genetic, people would rather take MC4 or NPY or leptin forward because of the huge genetic effect size in mice. But it doesn't work pharmacologically. So GLP-1 is involved in all these physiological processes, but it's a very tiny effect. And then because some brave clinicians just took this hormone and put it into people and found out that this was like the most efficacious of the two incretins back in the late 80s. That's how we actually got the first run at it, and understood that we probably shouldn't look at the mice, we should look at the human data.
Okay, that's what I was thinking, because Suad Efendi in Karolinska, he looked at it and he actually reported the slowing of the intestinal emptying as well. This was a fabulous paper too. So anyway,
he also did a lot of important work, right? And
Suad Efendi did that. We worked with Suad, Richard's lab. Anyway, thank you so much for that.
Another one of the giants that died before he saw the success, right?
So that, Thank you. Thank you for the opportunity. It was great.
Thank you so much, Sita. I appreciate it. Thank you. All right, now we have multiple people with their hands up and a lot of questions. Let's see what
I started a trend.
No worries, Lotte. I'm glad this is so interactive. There's a lot going on. But I do want to mention it's 12:05, and you're okay to stay a little longer so we
Yes,
can continue?
I'm,
Okay, wonderful.
you know, it's past 6 in so no one's at work anymore here, but I'm
All
happy to
right,
stay.
let's stay a little longer. So let's take one from the Q&A box, and maybe we can go back and take one from people who would like to speak. Let's see. Here's another question. Hi, Lotte, great talk. How important is biased agonism in your drug discovery pipeline when studying different GLP-1 receptor agonists?
Yeah, so semaglutide is not a biased GLP-1, right? I'm assuming we're talking about beta-arrestin bias, right? The semaglutide isn't biased, right? Then tirzepatide, which is a dual-acting GLP-1 GIP, is biased, but you know the efficacy is kind of like similar with the two molecules when it comes to weight loss. So I think it's just a little bit difficult to figure out, right? Definitely bias has been linked to a better weight loss and better glucose lowering, but it's not super clear from the clinical data because then, you know, I think orforglipron is also biased, but then that doesn't have better efficacy. But then that might be because of also limited by the physical chemical properties. It has a shorter half-life, right? So there are all of these things to take into consideration. You know, how does it activate the receptor, but also how long half-life does it have it? What's the on and off rate, right? So it's definitely a hypothesis, but for semaglutide, it's not involved with the efficacy. So you can get a really good efficacy without bias.
Fantastic. Thank you, Lotte. All right, Katerina, you can go ahead and ask your questions. Abigail, if you could unmute Katerina. I think, Katerina, you can unmute yourself now and ask your question.
Yes. Hello, thank you for the opportunity. And thank you for amazing talk, Lotte. I have a controversial question about like all the pluridimensional effects of GLP agonists or similar peptides. So do you think that could be actually all the functional effects might be through binding to different targets, and that, I mean, beyond the known GIPR or glucagon, and not just through GLP-1 receptor, for example? There are a lot of effects which are, for example, related to parotid hormone receptor. Do you think GLP could be binding to different GPCRs, and that the multifacet of their function would be like because of it and not just because of GLP binding GLP-1 receptor?
Yeah, so I get the question, and I would say no, because you know why would that be? You know, GLP-1 is a peptide that is highly involved in physiology. It's very specific. You know, you have to go to 1,000-fold higher doses in order to get binding to any other receptors. So I just don't see any data that supports that should be the case. Can I rule it out? You know, but I could, if you gave me 4 hours, I could take you through many, many more outcomes and describe exactly which GLP-1 receptor is activated on which cells and how that relates to the different outcomes. But in general, I would just say no, because, you know, it's a very specific binding to the GLP-1 receptor. And of course, when you get a medicine approved, you actually have to show that. So there's lots and lots of data in the files behind the approvals that go towards studying that there's no known binding within any kind of relevant range that hits other receptors.
Okay, cool. Thank you.
Thank you so much. Thank you, Lotte. Okay, so we're going to take 2 more questions from the Q&A box, and then we'll wrap this up. Any effects on fertility, on pregnancy?
Yes, obviously, when people lose weight, and some might also have PCOS, and it's highly advantageous to lose weight, and then people have an easier time getting pregnant. So we are actually all of the companies have had problems with people getting pregnant in the studies where they were not supposed to get pregnant during the clinical trials. So there are pregnancies, right? And there are no adverse outcomes described. There is, of course, because with the obesity approvals, there is a pregnancy registry that has to complete. And because, there's always a concern with pregnancy, you shouldn't lose weight, right? And there are also some animal data that have found changes in rats and mice, but then not in non-human primate. And there's a very good explanation for that about how the yolk sac in mice and rats express the GLP-1 receptor and actually completely surround the fetus, but that's not the case in higher order species.
Awesome. Thank you a lot. One more question and then we'll wrap this up. So we have a question here. It says, I worked on developing GLP-1 receptor allosteric modulators in grad school. Unfortunately, I was not successful. Do you think that advancements in cryo-EM structured guided drug design will make development of allosteric modulators for this receptor possible in the future?
Oh, that's a tough, open-ended question where I have to be an oracle to predict the future. It's remember the GLP-1 receptor is a GPCR of the B family, a fairly large exercise. So the peptide has to bind both the big hand outside the cell and as well as dock down into the receptor. But then, of course, I think the question goes towards, are there other binding sites that are not the orthosteric binding site that you could find? Yeah, it's definitely possible. And now with all the advancements in cryo-EM and virtual screening, right? It's a possibility, but it's super hard to predict the future. And I just want to say one thing, I'm so happy I didn't get any questions where people said GLP-1 inhibitors, because I hear people saying that. Some people think that it's inhibitors. This is why I had GLP-1 receptor agonist written out on many of my slides, just to point that out.
Awesome. Okay. We are 12 minutes past noon, so I think we have to stop. You have a lot of engagement, a lot of questions. Perhaps we'll bring you back to another webinar or bring you here at NCATS and we can continue these discussions.
Yeah, and people can write to me if they have important questions. Yeah.
And that too. So please do reach out to Lotte if you want to continue these discussions. Thank you so much, Lotte, for this wonderful presentation, all the time you took to answer all these questions. And again, for all the efforts that you've done to bring this medicine to people in need. We really appreciate you. And thanks our listeners for hanging in, asking so many interesting questions. The Q3 webinar will be by Dr. Ben Cravat. We're really excited to have him next. So thank you, everyone. You all have a good evening, morning, wherever you are in this world. All right. Thank you. Bye-bye.